I'm really excited today to introduce Dr. Donald Ingber. We have a nice presentation scheduled, and what we will do is have him speak first and save questions for the end. So you're welcome to pop them in the chat as we go, but there'll be time for you to raise your hand and ask them at the end as well. So I'll give the introduction and then we'll kick off. So Dr. Donald Ingber is the founding director of the WIST Institute for Biologically Inspired Engineering at Harvard University. He is a pioneer in the field of biological inspired engineering, and at the WIST Institute, he currently leads scientific and engineering teams that cross a broad range of disciplines to develop and commercialize breakthrough technologies to advance healthcare and improve sustainability. He is a member of the National Academies of Medicine, Engineering, and Inventors, American Institute for Medical and Biological Engineering, and the American Academy of Arts and Sciences. We are so excited to have him here today to speak to us about human organ chips as replacements for animal testing. Thank you very much, and I will hand it over to you.
What I'd like to do today is describe work that we've done over the past 15 to 20 years now in terms of the development of human organ chips as replacements for animal testing. I'm going to give you a broad overview that goes beyond immunosafety, but I think it's important to see the range of model systems and the range of what we can measure, because we kind of integrate immune cells in many of these different systems. So organs on chips are basically devices that we originally created with microchip manufacturing because they have control over features at the nanometer to micrometer scale that we line with living human cells, and our chips are enable us to reconstitute organ level structures and organ level functions, not cell or tissue, but organ level. And the idea was that this could accelerate drug development, replace animal testing, and advance personalized medicine. And I'm going to give you examples of all that today. So our first breakthrough was now 16 years ago in science. We called it a human breathing lung on a chip. We're not trying to build, a whole organ. You have to think of these as living three-dimensional cross-sections through a major functional unit of an organ. And we started with the air sac or alveolus, and we call that a lung alveolus chip now. And as you probably know, the alveolus is where we have gas exchange, aerosol-based drug delivery, pneumonia, COVID, et cetera. At the EM level, at the bottom right, in a cross-section, it's a simple structure. There's air. There's one layer of alveolar epithelial cell, flexible, porous basement membrane, extracellular matrix, capillary cell, and then blood. What it doesn't show you is that this is an incredibly mechanically active structure. And we know from respiratory physiology that the breathing motions are extremely important for both physiology and pathophysiology. So if you're trying to distill down, to the simplest design principles, like what makes an organ, it's usually two or more different tissue types that interface. New functions emerge. Almost always one of them is a blood vessel to oxygen, nutrients, remove waste, etc. And I've worked for 50 years now in the idea that mechanical forces are as important as chemicals and genes, so that these mechanical forces are important. So we wanted to basically have tissue-tissue interface. We want to have dynamic flow to mimic vascular perfusion, and also those physical forces due to shear stress, and then cyclic breathing motions. And so the next animation shows you how this works. At the top right is the chip. It's the size of a thumb drive. It's optically clear, made out of a flexible silicone rubber. If you cut it in cross-section, there are 3 hollow channels, each less than 1 millimeter wide. The middle channel is cut into top and bottom by a porous membrane, and that is coated with extracellular matrix. To do the lung chip, we have lung alveolar epithelial cells on top, human capillary cells on the bottom from lung. We just recreated the alveolar capillary interface. And then the trick is that we apply cyclic suction through side chambers and it's all flexible, so it stretches and relaxes at the same rate as when we breathe in and out. We could then flow air over the top to create an air-liquid interface, and we could flow medium with or without immune cells through the bottom channel, or whole blood for short times, as I'll show you. Now, if this were to work, it should mimic an organ level response. Imagine you have a bacterial infection. There's a tissue signaling response, and that the epithelial cells put out inflammatory cytokines. They activate the endothelium, which usually just has the white blood cells rolling by. When they get activated, they express ICAM1 and other adhesion receptors in the cells, stick, roll, migrate, and clear. That's homeostasis. So now this is imaging in the device. Those are fluorescently labeled fresh neutrophils that are flowing through. You don't see the endothelium because it's unlabeled, and the epithelium is behind the screen. So to begin with, it's a healthy endothelium. They just flow by. We now put bacteria or cytokines on the other side. Now you see them being pulled out under flow, which is that first step is a shear stress dependent adhesion step. And you see them attaching to the endothelium. And now I'm going to go to higher mag, and you see one neutrophil that right about now goes between two unlabeled endothelial cells, migrates through the matrix-filled pore, goes out of focus as it goes to the epithelial side. Now by phase, you see it coming out of the epithelium. Now I'm going to show you the bacteria in GFP green, so the immune cells are in red, and you watch them in being engulfed. So you just watch the entire inflammatory response in a human lung at a high resolution in this little rubber device. So we were funded at the time to look at nanotoxicology, the safety or lack thereof of immunoparticles getting into our bodies. The lung was an obvious location. We used simulants of smog, air nanoparticulates, silica nanoparticles. We used fluorescent reporters to look at reactive oxygen species release, free radicals. And what was interesting is that when we added these nanoparticles, we saw a time-dependent increase in reactive oxygen species, as well as recruitment of immune cells, but only when we had the physiological breathing motions. If you did this in a transwell with the same cells and medium or static chip, you do not see this. So this would have never been seen. We could also quantify absorption of these nanoparticles across the tissue-tissue interface by just collecting the effluents, the outflows of each channel, which we can collect separately. And what we found is with breathing motions, we got an 8 to 10-fold increase in absorption of these particles. This is not due to tearing of the tissues, because if you give fluorescent albumin or dextran, there's no difference. This is a mechanically induced transcytosis response across two different cell layers. Now, this was not mimicking a clinical response. This was a prediction. No one had ever seen it before. So we actually developed an ex vivo ventilation perfusion model on a mouse where we could nebulize the particles while controlling breathing with a ventilator, and we got the exact same response. And that was published as a full article in Science, and this really opened the organ chip field, showing that this is really a possibility. So I can't show you everything in an hour because we have done probably 20 different organ chips, healthy and diseased, of pretty much any organ you can think of. But what I do want to go quickly is we have shown examples of the value of these chips for drug toxicity testing, modeling human acute and chronic diseases, modeling drug efficacy and development of drug resistance in clinically relevant contexts, drug discovery, AI-enabled drug repurposing, vaccine safety and efficacy, food, nutrient, probiotic testing, assisting in clinical trials design, and even personalized medicine. I won't be able to do everything, but I'm going to give you sort of a Disneyland ride through a lot of this. So I know you're interested in toxicity testing. Our first lung chip that I showed you, we followed up with a model where we modeled interleukin-2 induced toxicity. Dose-limiting toxicity is pulmonary vascular leakage leading to pulmonary edema. It's also a disease model. We perfused IL-2 at a clinically relevant dose through the vascular channel. It's an IV drug. Time 0 looking from above with air, that's what the epithelium looks like. And over 2 to 4 days, you saw a meniscus of fluid filling the airspace, which is the exact same time course as seen in patients. When we quantified this, once again, breathing was absolutely critical to see this response. The red squares are without breathing. And we confirmed in the animal model that this is actually true in vivo as well. Because I've worked on mechanobiology so long, I had identified one of the earliest signal transduction pathways in terms of when cells feel a stretch, and that's the TRPV4 ion channel. And I heard GlaxoSmithKline was working on an inhibitor, and we tested that, and we completely knocked out this pulmonary vascular leakage, pulmonary edema in our human chip model. And they did the same in dog and I think mouse cardiogenic pulmonary edema models. We had back-to-back papers in Science Translation Medicine, and that drug moved on to clinical trials. Now, I mentioned we could perfuse these chips with whole blood. If you have endothelium on all four sides of the bottom channel, they are a natural anticoagulant. And so here is whole human blood with no anticoagulants, and you're watching the cells move through by phase contrast. Now, we can quantify platelet endothelial dynamics and clot formation in these chips by using fluorescent fibrinogen and fluorescent platelets. And we've done many studies on this, but just one that I think is really relevant is that we tested a therapeutic antibody, a biologic that was against anti-CD4L that went through animal studies without any toxicity and actually had thrombotic disasters in phase 1 clinical trials, pulmonary thrombosis, cardiogenic problems. And that was not seen in animal studies, but when we tested this on our chip, we actually could mimic that thrombotic toxicity. And we actually could show it was only with soluble CD40L, and the companies later discovered that and modified the FC portion so that they can get around that. But the point was that you could see this on chip. We developed a bone marrow chip where we take CD34 cells from blood or marrow from humans. We put them in a matrix gel, filling one channel with bone marrow-derived stromal cells. Then we put endothelial cells in the second channel, and we only perfuse through the vascular channel, just like in vivo. And as you can see from the FACS data, fluorescent cell sorting data, that you get near normal neutrophil and erythroid differentiation over a month in vitro in these chips. And this is a Guim sustain showing you get multiple different hematopoietic cell types. Now with this, we actually replicated 5-fluorouracil toxicity. It's well known that it causes bone marrow toxicity. It's been shown in vitro in marrow cultures for years, but in vitro, people have to bathe the cells at static culture in a high dose for long times to see marrow toxicity. What we can do is we mimic the clinical pharmacokinetics. So because we have perfusion, we give a 2-day infusion of about 4 micromolar, which is precisely what you see in patients. And there we can detect the bone marrow toxicity where you don't see it in suspension cultures or static gels. On the right, each dot is a different patient to give you a feel for the reproducibility of these results, which was extremely good in the patient. Each chip is made with CD34 cells from a different patient. AstraZeneca saw that and came to us and asked if we could help model a very peculiar regimen-specific toxicity that they had seen in phase 1 clinical trials with a cancer drug. They saw a 2-hour dose gave neutropenia and anemia, 2-day dose gave neutropenia alone, and they couldn't really replicate this in animal studies at the time. They had the pharmacokinetics they measured in the plasma of patients in the trial. That's in gray at the top right. Because we have flow, we could precisely mimic that PK. When we do that, we mimic that regimen-specific toxicity. Now, I think you probably have heard about this, but the real breakthrough is our liver chips and what we've done demonstrating their ability to model drug-induced liver injury better than animal models. So these chips have 4 different primary human cell types. Hepatocytes in a sandwich of matrigel in the top channel, and then liver sinusoidal endothelial cells, Kupffer cells, and stellate cells in their correct positions in the bottom channel. And we showed in this paper that you recapitulate liver structure and function. These are biocanaliculi at the top right pictures. This is albumin and urea production, here shown over a week, although we've done multiple weeks. Liver-specific gene expression relative to day 0 primary hepatocytes when they're, these are frozen, and when they're defrosted, and this is over days in culture. And at the top, and at the right, we looked at multiple drugs alone at toxicity, looking at ALT and albumin production, and also morphology, again, mimicking the response seen in vivo. But the real study that we did was we tested 27 different drugs, most of which were identified by the Pharma IQ Consortium as sort of benchmarks where we knew the results in animals and humans, and most of them were wrong based on animal results. We tested 870 human liver chips with cells from 3 different human donors. Many of these toxicities had been missed before, as I said, and these chips were able to predict human small molecule drug induced liver injury, DILI, with 87% sensitivity and 100% specificity. And I personally think we would have gotten even better if we had immune cells in these chips, but we did not in this study. That was 7 to 8 times better than animal models at predicting human DILI. And we had an economic analysis in this paper which basically estimated that the pharma industry would save $2 to $3 billion a year based on replacing animal drug-induced liver injury data with this test by preventing late-stage failures in the pipeline. I should also note that Moderna has publicly disclosed, and allows me to show this this slide, that they've been using the liver chip, the human liver chip, instead of non-human primates. They use transcriptomic analysis of these chips because they're using these internally to look for liver toxicities, fibrotic toxicities induced by lipid nanoparticles for their RNA therapeutics. And this just shows you that they found that it was as good as, if not better, because it was human, than the non-human primates, but they were able to do these studies in 1/4 the time and at 1/10 the cost. And I really hope this is what changes how industry does this in the future. Now, just to give you examples of more broad, impact, modeling diseases, efficacy, and resistance. We had companies come to us and they were interested in drugs for COPD and asthma, and they said the alveolus chip wasn't what they were looking for. They were looking for, a small airway, a large airway. So here is a lung airway chip with bronchial epithelium, human lung endothelium. You get beautiful cilia. They move in a directional way. If you put fluorescent microparticles to look at mucociliary clearance at the bottom right, that's at the same rate as in all of our lungs at this very moment. We actually did cigarette smoke exposure. This is a cigarette smoking robot. That's a cigarette lighter from a car, a Gatling gun with real cigarettes, and it mimics the breathing pulses and pauses and intervals, and real smoke goes to the airspace of the chip, not extract. And this is just one result from that study. If you look at IL-8 as a measure for inflammation as a toxicity, and you look at smoke exposure in a healthy chip, we didn't really see it. But if you have chips with cells from patients with chronic obstructive pulmonary disease, and you're looking at comorbidity effectively, we double IL-8. And at the right, you're seeing transcriptomic results showing changes in gene expression after smoke exposure in healthy chips compared to before. At the left 9 lanes, that's the right 3 lanes. The nine lanes at the left are from a past clinical study of healthy people who smoke cigarettes, where they did transcriptomic analysis. We were very excited. The top quarter versus bottom 3/4 are very similar up and down. Reviewers didn't like it because ours were too similar to each other compared to real patients. Then we realized that we could do this type of study better than a clinical study because it's matched comparative modeling of the same patient before and after stimulus. Those gene changes are due to smoke, whereas most of the changes are due to smoke exposure in the patients. There's also differences in family history, work environment, home environment, et cetera. And so we actually then got accepted and we're asked to do a mini review on comparative modeling in vitro. Cystic Fibrosis Foundation gave us cells and asked could we model cystic fibrosis in these chips. As you can see at the top, those are the full length of the channel, and you get many more cilia in green, which is what you see in patients. Cilia beating frequency is higher on the chips at the bottom left, not seen in a static transwell. More mucus production, major problem in these patients. More baseline immune cell recruitment in these chips, and inflammatory cytokine release. And now we can study microbiome. So if we put Pseudomonas aeruginosa, which is in all of our lungs, but terribly overgrows in these patients, we see terrible overgrowth on the chips. We've also developed influenza infection model. NIH and DARPA asked us to do this in 2017 because they were worried about potential pandemics, but they were worried about influenza at the time. It's a beautiful model of viral infection. You see GFP virus infecting the cells, you lose junction, Z01, cilia, you even see endothelial injury that most people don't study when they just do epithelial cells in culture. When you use three different viral strains, H1N1, H3N2, H5N1, you get different virulence seen, much like seen in the patients. And very importantly, you could study the effects of comorbidities, which often lead to failures in clinical trials, but you don't study in animal models. So when we look at COPD chips, COPD patients are much more sensitive to exacerbation by influenza. We get a tenfold, a whole log higher viral load and much higher inflammatory cytokines. And we can nicely measure the effluence of these chips. So we can measure, this is a cytokine storm, if you like, and we did the different viral strains, and the cytokine levels, this is 6 different cytokines, matched very much what has been seen in vivo in patients. Again, you could watch immune cell recruitment like that movie I showed you and watch clearance of virus. And now you could study drug effects. So we perfused with oseltamivir, Tamiflu. You probably know the FDA has approved this as the first-line drug for influenza, but it's only approved for the first 48 hours after infection because it loses activity in patients, and that's exactly what we saw on chips. We did an interesting study where we did influenza infection plus or minus breathing motions, and we found that breathing suppresses infection and cytokine release dramatically. And don't have time to go into it, but using transcriptomics, proteomics, we found that breathing activates innate immunity. It increases the production of protective type 1 and 3 interferons. When we did transcriptomic analysis of breathing versus non-breathing, this gene S100A7 came way up, and we confirmed it at the protein level, and that is a ligand for the receptor for advanced glycation end product, RAGE, which is highly expressed in lung cells and is a major mediator of inflammation and inflammatory side effects in different diseases. We found an existing drug, azilurigon, that is a RAGE inhibitor that was in phase 3 clinical trials, oral drug, well tolerated. Clinical trials for Alzheimer. It completely knocked out the cytokine storm in these chips. These very data were included in a pre-IND application to the FDA by Cantex Pharmaceuticals, and that drug is now in COVID-19 trials. Recently, we've developed a double-stranded RNA therapeutic that actually induces type 1 and 3 interferons. And with ARPA-H funding, we're looking at it as a treatment for cancers. It works beautifully with viral infections. And we have a cancer chip where we take human non-small cell lung cancer cells labeled with GFP, and we put them at low density in a healthy chip. And we watch their growth in a normal human lung microenvironment. And in this study, we're testing this RNA, but we're delivering it with lipid nanoparticles. And we actually can now look at both lipid nanoparticle toxicities and liver chips versus lung chips, and then the effects with lipid nanoparticles and therapeutics. This is showing you a potent dose-dependent response. In this case, the lipid nanoparticle alone is not toxic, but we can see the dose-dependent effect of this anti-cancer immunostimulator. We developed human intestine chips. We now take organoid biopsies from patients from intestine, make organoids, grow them in 3D gels the way organoids are grown. The problem is with organoids, they grow beautifully and we expand cells that way, but you can't get access to the lumen easily. You can't look at microbiome, absorption, transport. So we break up the organoids, plate them on the top channel, and we put endothelial cells or stromal cells on the bottom channel, and you get absolutely beautiful intestinal villi forming on these chips, but now you could get access to top and bottom. Now, this paper is in press in Nature Biomedical Engineering, and it's kind of really amazing. We get not only the organoids from the biopsy, but the stromal fibroblasts from the same patient biopsy. And then we reconstitute the epithelial stromal interface on chip. And when we get these from patients with inflammatory bowel disease, either Crohn's disease, CD, or ulcerative colitis, we make the chips. You can see that we reconstitute the morphological changes, the flattening of the of the villi or crypts, the columnar cells becoming more cuboidal. You see increased inflammatory cytokines. If you look at mucus production, we can do this by turning the chip on the side and do dark field imaging in live cultures. You actually see the thinning. This is the top versus the bottom, or quantified here. Thinning of mucus in the IBD chips, as you see in patients. In this study, we found that the peristalsis-like motions, which we mimic here, actually regulate mucus production, and it does it in healthy and IBD, but there's still less in IBD chips. Interestingly, we have a stroma, we can look at fibrosis, and we can use second harmonic imaging, and we find that, well, there's a little bit more fibrosis under static conditions in IBD chips. When you mimic the full peristalsis, you get rampant fibrosis on these chips. This is an interesting subtlety in, if you like, safety, or in this case, disease modeling. It's well known that women who have IBD have to get the disease under control because they could have terrible exacerbations. So we made female IBD chips and exposed them to pregnancy hormones. And what you see are greatly increased inflammation and incredibly increased fibrosis, which may be part of why they have such a painful condition. But the most interesting thing is that we could do tissue recombinants in these chips. We could do healthy epithelium with IBD stroma, IBD epithelium with healthy stroma, or homotypic recombinants. What we find is that the IBD stroma is the major driver of inflammation. And this may be important for toxicities in the future as well. It's the major driver of inflammatory cytokines. It disrupts the barrier. And when you look at circulating immune cells, if you have IBD chips with both epithelium and stroma, you have a high recruitment into both channels. But if you have a healthy stroma with IBD, you lose that. And same with healthy chips. So the stroma is inducing changes in the epithelium that are a major part of this disease. All our drugs target immune cells and epithelial cells, so a whole new area for drug development. The most surprising things to us is that it's well known that IBD patients have a higher propensity for cancer formation. So we actually gave a carcinogen, ENU, to these chips for 3 weeks. We get flattening of the epithelium, much higher levels of inflammation. We get early stages of cancer formation or carcinogenesis as measured by downregulation of E-cadherin, upregulation of B-catenin, higher expression of CEACAM5. This is also enhanced by the IBD stroma being present. But we did Salo-Pass genome sequencing, and only the IBD chips express mutations and gene duplications. So we're really being able to model the early stages of cancer formation on these chips. Different area of food probiotic testing. Gates Foundation saw our intestine chips, said, could we model environmental enteric dysfunction? This is multifactorial disease, nutritional deficiency in low-resource nations, dysbiosis, malnutrition, pathogen exposure, immune system, all could contribute. We modeled this on chip by using niacin and nicotinamide and tryptophan deficiency, so nutrient-deficient medium. When you do that, you get villus blunting as seen in these patients in both healthy and EED chips, but you get much higher reduction in absorption of nutrients, fatty acid, peptides, amino acids, not shown here, and that's higher in these EED-derived organoid chips plus nutritional deficiency. At the right, we were lucky to have the transcriptome from duodenal biopsies of these patients, and what you see here are the gene expression. The left lane, all the way at the left, is the clinical data of genes up in EED patients versus down at the bottom. And I won't go into all the data, but the second lane from the left that most closely mimics it, with reds at the top and more blues at the bottom, is only when we have chips lined by cells from ED patients, but with nutritional deficiency. If you have the chips alone in full medium, you don't mimic it. If you have nutritional deficiency in healthy chips, you don't mimic it. You need both, probably epigenetic signature in these patients plus the environment. And then finally, microbiome is known to be important. So now we got EED microbiome from these kids that were passaged in Jeff Gordon's lab in mice. And now when you combine them, you get much higher inflammation and higher disruption of barrier. Point is, you could discriminate between the contributors, multiple different factors that contribute to a phenotype, whether it's disease or toxicity. Gates Foundation asks, could we make a human vagina chip? And we've now made vagina, cervix, fallopian tube, and we're doing uterus. But they were interested in testing live biotherapeutic products in these for bacterial vaginosis. We got epithelial and stromal cells, get a beautiful vaginal organ-like, tissue-like structures. And we could show if you put a dysbiotic bad microbiome, vaginal microbiome, you get high inflammation. If you give a healthy L. crispatus microbiome that is seen in healthy women, you suppress inflammation. And we actually could test live biotherapeutic products that went to clinical trials, and we got very similar results on these chips. Now, for the immunotox area, I think the work we've done with lymphoid organ chips is probably most interesting. We've made a lymphoid organ chip that is very simple. We get B and T cells from apheresis collars, primary cells, peripheral blood, but we put them at high density in matrigel in one channel, and we just flow medium in the second channel. And what we see is if we give a vaccine like Fluzone, influenza vaccine, we see spontaneously self-assembly of germinal center-like structures that have B and T cells, AID expressing, plasma cells are forming, and we get high affinity IgG against the specific antigen when we also include dendritic cells in these chips. If we measure the effluence, we're lucky to have data from patients where from immunized with influenza vaccine. We get very similar profiles of cytokines. The red at the top are on chip. I'm sorry, the ones at the right are on chip. The data on the left are from patients. We've used this for adjuvant testing. We've confirmed there's somatic hypermutation going on in these chips. More recently, today, a publication came out in Nature Biomedical Engineering, working with William Shee's lab at the Beast. They developed a very novel DNA nanostructured, self-assembling, huge DNA nanostructured cube that can position antigens at specific nanometer spacing. They have both CpG adjuvant on here and the antigen. I won't go into it, but they can show that the spacing of these of the adjuvant and the antigen actually determines the level of vaccination response. But here it's just showing using our chip, they did this in mice, but we did this in human with hepatitis B surface antigen. You get beautiful antigen-specific T cell activation, CD4, CD8, and multifunctional. They did it then with a full-length SARS-CoV-2 spike protein, and you get protection against many different variants, so a very broad spectrum using this. With GlaxoSmithKline, they asked us to test self-amplifying RNA vaccines. Those are usually injected intramuscularly where there's a monocyte response. So we basically made a muscle culture module, like we add the vaccine to the muscle culture with immune cells. We pass the medium and immune cells into our lymphoid organ chip, and we have beautiful mimicry of, replication of production of antibody response. This is a log scale with antibody production with the self-amplifying RNA vaccine that has a COVID spike antigen. And then in a paper that's in review, all the stuff I've shown so far are recall responses. We've actually shown that we can use a naive antigen to vaccinate. We used de novo antigen-specific IgG production against rabies, and we made LN chips with cells that we were able to obtain from donors who've shown to be naive, and we can show beautiful anti-rabies IgG production as well as cytokine production in the effluence of these chips. We're also now using this to model diseases. So we infect the lymphoid organ chip with live Borrelia burgdorferi bacteria that cause Lyme disease. And we actually really find that we suppress germinal center-like structure formation and AID expression, and we actually we don't get any T or B cell activation. So you're actually getting sort of an autoimmune response on these chips. And we've been using this, actually, we think we may have a new therapeutic target for this disease. Lastly, we were originally funded by DARPA with the idea of creating a human body on chips, like the idea of linking many organs together and combining it with computational PK/PD to shortcut prediction of human PK parameters to shortcut clinical trials design. And we actually have done this. We simplified it to a first pass model of a gut, liver, kidney chip. We have a AV mixing reservoir so that you could take samples from it that would be like phlebotomy of a peripheral vein rather than the outflow of an organ chip. We gave an oral drug, nicotine, to the lumen of the gut chip. We used mass spec to measure its level and its metabolites in every chamber. And then we collaborated with CFDRC that does computational PKD for pharma, so they know blood flow rates and tissue masses of every organ in the body, and they can scale from our little chips to in vivo. We then did an IV drug, cisplatin. We inject that in the AV reservoir. We don't need the gut chip, so we have a bone marrow chip to look at pharmacodynamics and liver, kidney. And at the rate, the colored dotted lines are predictions of PK from these chips with data and this computational analysis. The dots with error bars from past clinical studies. The top one was a Swedish study where they compared chewing tobacco, they call snus or gum, at 3 different doses, and our PK data precisely predict their, T1/2 Cmax. The bottom is from a clinical study with 2 different infusion rates with cisplatin. And again, same model, beautiful prediction. And then finally, this year, a year ago, in 2025, we published a clinical study with a collaborator who's a cancer surgeon at McGill. He has patients with esophageal cancer. They're treated with multiple drug regimens. They almost always become resistant. He has to choose a new combination. He has to kind of wing it. So he's been collecting biopsies and making organoids of the epithelial tumor cells and collecting the stromal fibroblasts. We recreate these on chips. And now the first thing is at the bottom, on the right side, bottom left, the whole exome sequencing. It's well known that organoids replicate the mutational phenotype of the cancer better than 2D cultures. But what we found is the chips are even better at replicating the mutational phenotype than the organoids when you have stroma and flow. Most importantly, if you expose these chips to a clinically relevant drug combo with a clinically relevant pharmacokinetics, same time exposure, if you give it to organoids, they die. If you perfuse it through the channel that would be going through the interstitium, through the stroma to the epithelium on chip, you beautifully predict patient sensitivity and resistant responses. And he's now using this to choose drugs for his patients. So to end, I really think the organ chips have the ability to change the paradigm in drug development. Now drug companies will spend hundreds of millions to develop it, tens of millions on a big clinical trial, almost always fail. Then they'll go back, do statistical analysis. Maybe they'll find a genetic subgroup that responded better. They'll 2 years later, 3 years later, do a small trial. If it works, they get approved for a now application. With, patient-derived stem cells, organoids, iPS cells, primary cells, you can make chips and flip the paradigm on its head. You could, have 50, 75, people have very similar phenotype, women, certain age, IBD, certain genetic subtype, find drugs that work in 80% of those patients and then test their other organ chips for toxicity, find the subgroup that are safe and effective, do a small trial. I think the likelihood of success is higher, the cost is lower, the time is shorter. And this, hopefully is what we do in the future. So with that, I will end. I have to disclose as a Harvard faculty member, you probably can't believe anything I say because I'm a member of the board, just chair the SAB and hold equity in Emulate. Now, Emulate has instruments and chips in almost every major pharma company, and it's in academic labs and companies around the world. The instrument at the left is called the Zoe. It cultures 12 chips at a time. That is what has been used for the last 10 years, but they are just about, they're completing the beta testing of the instrument at the right. The one at the left has 12 chips at a time. You have to put it in an incubator. You have to take the chips out to look at it on a microscope. The instrument on the right is the size of a Hewlett-Packard printer. It's called the AVA. It does 96 chips simultaneously. It's the incubator. It's a fluorescent microscope. You could do real-time imaging, and it's set up to hook up with robotics. And I think that is really where the future lies. And I hope this really represents a major turning point for this field. So with that, I couldn't do what I do without the incredible range of expertise at the Wyss Institute, and invite you to the website if you'd like to learn more. Happy to answer questions.
Thank you very much for that overview. It was a lot of work and lots of really nice intersections between fields. So thank you for that. There are some questions in the chat, and I would invite folks to raise their hands if you would like to answer. I'm happy to start with one from the chat. So I think there's actually two related to the DILI system you presented. Were drugs that were negative in this assay, positive for tox in human, assessed further to try to understand why this system doesn't pick up the tox?
There were only a couple, and Emulate did that study primarily at their site, and I don't, this is what's being gone through FDA's iStand program and has passed through all the stages. As two companies are testing the product now. If they get similar results, it will get approved that any company could use it in lieu of rat or dog preclinical DILI data. The way it'll probably be used in the near term is more as when you get conflicting results in those two species, but the answer is I don't know if they followed it up. I can just say that there were not immune cells in those chips, and I would not be surprised if that's what was lacking and why they didn't pick those up.
Yeah, understood. The second part of that question was if there were common attributes among those drugs that were tested.
We published a paper before that where we did rat, dog, and human liver chips, all with 4 primary cells from each species, and we showed that we can mimic fibrosis, steatosis, basically all forms of liver toxicity. So they covered a range.
Thank you. Another DILI question. You mentioned that your study lacked cells that drive adaptive immune responses, which is thought to contribute to idiosyncratic DILI responses. Is there progress in the co-culture systems that integrate that adaptive arm of the immune system?
We've been linking, first of all, you can, I didn't even describe that we've integrated alveolar macrophages in the lung chip, and dendritic cells into the intestine chip, and we've linked our lymphoid organ chip to other chips fluidically. So that's beginning to be done. I haven't worked on the liver chip. We're just starting it again now. I originally developed it in my lab, so Emulate has really done that. I'm pretty sure, it's not a problem to flow immune cells through the liver chip. I just don't know if they've done it with any of these drugs.
Yeah, okay. Moving on to IBD fibrosis. How long do these chips last in culture versus how long does fibrosis take to occur?
Well, we're seeing that fibrosis occur within, like weeks, days to weeks. The intestine chips are very robust. We can keep them going for, it takes 3 weeks to differentiate them, 2 to 3 weeks, and then we could keep them for at least a month or two. I should note that our bone marrow chips are going on the Artemis mission to the moon with the astronaut cells in them, because they're very robust, and those, as I showed you, last at least a month. The lung chips are harder. Because there's an air, it's easier to get that interface to tear. So those, we usually do for like 2 weeks after we differentiate them.
Great. Another question asks, what is the regulatory status of any of these models? Are you approaching a method or device qualification?
So as I said, the liver chip is the farthest along of any NAM that I know of in terms of a cell-based NAM. And that has gone through all the stages where it just needs to be tested by two commercial groups that get the same results. Yeah, that's it's that's 870 chips. You can imagine how the kind of level of study that the FDA requires to qualify. But hopefully that will get easier over time.
Yeah, that's a big question a lot of our groups are facing now.
I
So
should
I'm
note
glad.
that I've been funded by the FDA continuously since 2012. And so we've also made non-human primate chips I didn't describe. We've made
Oh,
lymphoid organ chips and lung chips that look really good, that are both similar but different.
interesting. All right, I'm happy to keep going from the chat, but I will also encourage folks to raise their hand and come on camera to ask them yourselves. But until then, I'll keep going. Do you use primary cells from the same donor to build your organ on the chip?
So the IBD were from the same donor. If they're asking about immune cells, we have not. We with the lymphoid organ chip, we're doing acute and chronic Lyme patients, for example. But when you're looking if you use peripheral mononuclear cells, we don't have a problem with crossing different donors.
All right, for lung on a chip, the importance of, where we go, fibroblasts was demonstrated in other organ systems. Will fibroblasts be introduced into the lung model?
Yeah, I mean, that's a really good point. It's not like there's one lung model, right? You have to always design these for a specific context of use, and the FDA is always emphasizing that, right? And our initial model was pretty good for pulmonary edema, but you can't use it for pulmonary fibrosis because you need the fibroblasts, right? So, I mean, we have in a later paper integrated fibroblasts as well. So you can do that. I often describe this as synthetic biology at the cell, tissue, or organ level. You start as simple as you can and ask, can you get clinical mimicry? If you can mimic clinical results, maybe it's simpler than we all thought, and you don't need to have all the complexity there. If you can't, you're missing something, and the fibrosis is a good example. So there we had to add back fibroblasts, but you definitely can do that.
Mm-hmm.
I also should note that we created a chip years ago for skin that is the same chip, but it's an open top and it's thicker on top, and it's more. It has flow, it has deformation capabilities, but it looks more like a transwell oval or round shape, and you could put a thick gel in it. So, like for skin, we had endothelium at the bottom, stroma with dermal fibroblasts, and then we had epidermal cells on top. And then you could have air at the top, you could put cosmetics on it or whatever. And so I think other people have used that to make models that have a thicker stroma to look at fibrosis, cancer models. We're doing a uterus right now. Believe it or not, we're funded to model heavy menstrual bleeding on a chip.
Wow.
And so we have not only do we have fibroblasts in the stroma, we have a microvasculature that links up to the bottom channel. So, and we could perfuse blood through it. So, so you can do quite complex things.
That's really interesting. The chip used for the naive immune responses to rabies, was this the lymphoid organ chip or a muscle chip? And was this done using rabies virus?
It was using rabies virus antigen. It was done with it was a self-amplifying RNA vaccine. So it had the muscle module where we have muscle cells and monocytes, and then we take the medium and the cells that have been activated in that context, and we move it to the lymphoid organ chip. And then the lymphoid organ chip we built with cells from a donor we were able to get commercially that were known to be naive against rabies, or confirm, and then we concentrated the memory B cells as well in that study.
All right. Would like to have your expert advice on the state of the art for early developmental exposure studies using the NAMS model?
For early development, that's interesting. Um, Dan Ho is my postdoc who developed the lung chip, then now has his lab, he's a full faculty member now at Penn. He developed a placenta chip a few years ago. I'm not sure how much is done there. We have a fallopian tube chip where we're looking at non-hormonal contraceptives, and we have human sperm going up. And there has been the thought of putting human eggs on there, but then we get into kind of ethical danger zone. I haven't done any early developmental stuff. I think people have done microfluidics with embryoid bodies and things like that and gastrulation, but I haven't seen anything done yet. But it's an interesting question.
Yeah.
And if we have a uterus chip, we probably can get, some of these, we can get things to implant. That would be quite interesting.
Right. All right. One last question here in the chat. Thank you for providing information on chips related to infectious disease prevention vaccines that primarily involve humoral immunity antibodies. However, is it feasible to create chips for cancer vaccines based on cell-mediated immunity that exhibit HLA dependency?
The HLA, if we can get, we're doing cancer chips now, and we are trying to get patient-derived cancer cells. And the question is, can we get immune cells from the same patient? That study where the esophageal cancer surgeon was getting organoids and stroma from biopsies, I believe he's also collecting patient-derived immune cells. So you need to do the HLA matching for those, and so it really depends on whether you can source the cells. That's for all these studies. Really depends on the cell sourcing.
Yeah.
You may need an IRB to do it, from the beginning.
Right. Great. That's all the chat questions. Are there any others from the group? Yep. Jiro.
Hi. So thank you for the presentation. It was lovely. I'm just curious, kind of a really broad question. What would you say is like the biggest challenge in the field right now? I mean, I think you mentioned like cell sourcing just now, so like maybe that's one of them, right? But when it comes to like developing these models or like using them, do you see like any other relevant hurdles?
So, I mean, clearly, integrating this more with artificial intelligence is something that, is beginning to happen. Needing more, integrating more sensors into these chips. We've in the past, it's not in the commercial unit, but we've integrated electrodes to do transepithelial electrical resistance for permeability barriers. We've done oxygen, we've done lactate. Other people have put fluorescent reporters in. Now that at least the systems that we use, Emulate system, has continuous fluorescent imaging, where you go from chip to chip to chip, while it's being cultured. That opens up a huge amount of data for AI. I think that, morphological signatures of toxicity are used by Emulate for the liver chip, for example. So that I think could be really, really powerful. To make the chips, it's really the cell sourcing. We have not had a chip we could not make. And, usually we're able to do it in 6 to 9 months. Totally new, starting if we can get the cells. It's more the throughput of these has been the big frustration, and the cost. The newer, higher throughput instrument, the chips are cheaper, which is nice. But it's really the throughput. We could do 24, 36 at a time. The other thing is that these are like animal studies, and that there's a huge amount of data that comes out, right? So you have the effluents and cytokines, you have transcriptomics, you have imaging, you measure permeability factors, and so you have multiple cell types that need to be cultured before they put on there and timed. So you need staffing to do that, right? So the more that you can roboticize all this, I think it will get faster and faster. But it's usually the cell sourcing, the quality of the cells is absolutely most critical. Technically, you know, big problems are bubbles in microfluidics, and that emulate's been pretty good at getting around. And then, yeah, I mean, those are the big things, I think.
Thank you.
Thanks. Any other questions? Okay. Well, thank you so much, Don. We really appreciate the lots of information and taking the time to answer our questions. I guess I will close us out for today. Thank you again. Round of applause, and
My
I
pleasure.
hope everyone
Thanks.
has a good rest of your day.
Bye-bye, guys.
Bye all.